Have you ever wondered why you can get your flu shot, do everything right, and still end up with the sniffles a few months later? Well, welcome to today's Explainer. We are diving deep into the microscopic world of our lungs, right from an immunologist's perspective. Honestly, by the end of this, we're going to fundamentally change how you view your body's frontline defenses. Let's get right into it.
So think about this number, 154 million. That is the staggering number of lives that vaccines have saved over just the past 50 years. It's absolutely wild. And this monumental achievement, it's mostly thanks to our immune system's biological memory. We're talking specifically about memory B cells, these incredible little units that remember past invaders and literally leap into action the second they return.
But despite this massive, world-changing success, there is a catch. Think about it. Why is it that a standard arm injection can absolutely save you from severe, life-threatening disease, but it kind of struggles to stop a mild respiratory virus from just setting up shop in your nose or lungs? What's a disconnect there?
Section 1. Understanding memory B cells.
Okay, to understand this little vulnerability, we really have to look at two totally different types of defense. First, you have systemic immunity. This is driven by those classic intramuscular arm shots. Think of this like having state police patrolling your entire bloodstream to prevent a full-blown crisis. But then you have mucosal immunity. That's like stationing elite special forces right at the actual borders, like your respiratory tract. And the thing is, a standard shot in the arm really struggles to build up that local border garrison.
So the missing piece of the puzzle here is the BRM. That stands for tissue-resident memory B cells. These guys are your dedicated local defenders. Unlike the cells that just sort of float around circulating in your blood, BRMs, they never leave their post. They are stationed there. But to actually set up this non-moving defense, they need to encounter the threat locally, like right there in the tissue itself.
Section 2. The lung's dedicated defense force.
For the longest time, we used to think the lungs were just, passive waiting rooms for antibodies. But research from leading immunologists has completely flipped that idea on its head. It turns out the lung is actually a highly active training ground. It's this dynamic place where immune cells continuously adapt to airborne threats. And get this, they even generate brand new memory B cells right there on the spot. It's practically a military academy in your chest.
It helps to think of the lung as a dual-layered fortress. It utilizes two totally distinct microenvironments to defend you. On one hand, the alveoli act as your rapid border patrol, constantly sweeping a massive surface area. And on the other hand, you have structures known as eyebolt, which act as the deeply strategic, specialized command center.
Section 3. Alveoli and lymphoid microenvironments.
So, when a virus actually invades, these two areas coordinate this absolutely perfect 1-2 punch. First, the alveoli respond instantly. No need to wait for T-cell backup or anything. They just immediately pump out IgG antibodies. Meanwhile, the eyebolt response is slightly delayed because it relies on T-cells, but the trade-off is huge. It produces IgA, which is literally the gold standard for defending your mucosal surfaces.
Section 4. The precision strike of local antibodies.
Let's look at how that first strike actually happens. Upon reinfection, BRM cells in the alveoli become highly mobile. They are like heat-seeking missiles guided by macrophages. Triggered purely by the innate signals of the viral RNA itself, they just instantly unleash massive amounts of IgG antibodies. It is rapid, it is completely independent, and it is absolutely relentless against the virus.
But meanwhile, over in the organized eyeballed structures, a much more strategic game is unfolding. These BRM cells interact with specialized tissue-resident helper T cells, and through this close collaboration, they undergo further maturation to pump out sustained long-term levels of mucosal IgA and IgG. Basically, they are locking down the entire respiratory surface for the long haul.
Section 5. The future of intranasal vaccines. Because we now know the lungs absolutely require local stimulation to build up that permanent BRM garrison, intranasal vaccines are turning out to be a total game changer. By delivering the antigen directly into the upper airway, like through a nasal spray, we essentially mimic a natural infection. This local delivery actively recruits BRM cells and stimulates local IgA production, aiming for what we call true sterilizing immunity. That means stopping the virus cold before it ever even takes root.
Which brings us to one of the absolute coolest strategies in clinical development right now, the prime and spike approach. It works like this. First, we use a traditional arm injection to prime the body with robust systemic immunity. Then, a bit later, we spike it with an intranasal booster to build those dedicated local mucosal BRMs right in the lungs. It gives you the ultimate double-layered protection. Best of both worlds, right?
So, I'll leave you with this thought. If our lungs naturally possess the blueprint for this perfect, localized defense network, and we are finally learning exactly how to activate it, are we genuinely looking at the end of the era of the needle? Because honestly, this isn't just some neat scientific trick. It could very well be the next massive revolution in global public health. Thanks for joining me on this explainer, and we will catch you next time.